Okay, great. Thank you very much to the organizers for having me here. I hope to speak for the next hour or so about graph theoretic analysis. I'll structure it kind of similar to the last talk, where the first 20 or 30 slides or so, I'll cover theory, and then we can mess around in MATLAB a little bit, time permitting. I know that I'm the only thing standing between you and lunch, so I will keep that in mind. Obviously, let me know if you have any questions. Take a look at the link that was sent. I think it now lives on GitHub and on Dropbox, so we can hopefully learn something together. The jumping off point here is that really the neurons are the fundamental unit of the brain, the fundamental cell type. And neurons are engineered to receive information. So you can see right there through their very sort of bushy dendrites and their dendritic trees, they can receive and integrate information. They also can transmit information to other neurons via axons, so in the form of electrical impulses. The way that they're organized is clearly then going to be important for what they do collectively. They tend to form little neural circuits. They are then organized into larger columns, layers, and so on, and neuronal populations. The question that then we want to address is, how does the collective activity, how does the collective signaling of the system give rise to emergent phenomena like perception, cognition, and action? This sort of field of connectomics is a little bit unique in the sense that you can almost trace it to one particular opinion paper, where the authors argued that as with any biological system, in order to understand its function, you should know its structure. And they argued in that paper that systematically compiling a list of all the different neural elements and their connections should be a primary research objective, not just of individual labs, but agencies and entire sort of scientific programs. So this kind of the term that they came up with, the sort of connectome, was a direct analogy to the word genome. The ome part signifies that this should be a universal, comprehensive catalog of all possible elements and how they're interconnected with each other. And this has given rise to this field of network neuroscience that has sort of many different research questions that revolve around it, but some are more technological in nature. How do we actually map? How do we record neural activity? And some are more analytical in nature. So how do we actually analyze, model, represent the various connections and interactions between neurons? And one of the paradigms that has emerged, and that's what we'll talk about today, is this idea that it becomes very convenient to represent neural circuits as a graph, this mathematical object where we think of the cells or other kind of forms of larger bits of gray matter as the nodes in the network, and we think of the connections or the interactions between these nodes as the edges in the network. The nice thing about representing the brain this way is that it gives you a whole host of statistical measures that you can use to articulate and quantify different aspects of network topology and how it's actually organized. Just a little bit of history, because I think it's quite instructive. The way that the field started was when Leonard Euler, he lived in Königsberg, which I think is modern-day Kaliningrad. Königsberg has a couple of rivers, and they kind of divide the city up into several distinct land masses. And at the time, there were 7 bridges that went across the various rivers. And he wanted to know if there was some way that you could travel through the whole city using each bridge only once. And the insight that he had was that really, in order to solve this problem, you don't care about things like topography. You don't care about where the individual streets are located. You don't care about where the lights and the park benches are. What you care about are the landmasses in the bridges. So he said, well, maybe one way to represent this was to say that the landmasses would be the nodes of a network and the bridges would be the connections, the edges in this network. And by coming up with this graphical representation, he was able to show that it actually wasn't possible to do this. So the field really lay dormant for a couple hundred years until Erdős and Renyi came around, and what they were doing was to mess around with this idea of random graphs. They were placing nodes in space, and they were saying, well, I'm going to start putting connections with some degree of probability between pairs of nodes, just randomly placing them. And what they showed was that in this kind of simple paradigm, you could come up with analytical expressions for a large number of interesting things about these networks. So, for instance, one of the classic results is that the degree distribution of these random graphs, that is, so the degree is the number of connections that a node has that it participates in, and the degree distribution is then what are the degrees of all the nodes in the network. What they showed was that this degree distribution would be binomial, and if you had enough of these nodes, it would start to look like a Poisson distribution. And so people started, this was the first time where people finally had tools to look at real data sets and to make guesses about what the structure of various naturally occurring and man-made networks would look like. But the field really kind of took hold in around the year 2000, where there were 2 or 3 very influential papers that came out that really started to get people to think away from the concept of random graphs. One was by Barabási and Albert, who they first looked at these Erdős-Renyi random graphs, and what they said was, well, if you assume that all real-world networks are random graphs, you expect to see a degree distribution something like this. So these are the various degrees in the network, and this is their relative probability. They have these sort of exponentially truncated tails. That is, there are relatively few, if any, nodes that have very high degrees. There are relatively few hubs or extremely important nodes, and the reason for that is in a random graph, you're just randomly placing connections between areas. So there's no reason why one node should be particularly privileged relative to other nodes. But they said, well, and this was, like I said, around year 2000, so they actually finally had access to really good data in various sort of social networks and various other systems. And they said, well, in many real-world networks, like in which actors tend to co-star in similar movies and the World Wide Web and so on, these degree distributions actually do not have these truncated tails. They actually are, in log-log coordinates, they actually look more like a power law. There's a straight line that you can draw there. In other words, there exists some group of nodes down here that have very high degrees. There exist some hubs that are more important than the other nodes in the network. And they call these networks scale-free. The reason being that in a random graph, because there is a particular scale. You can kind of think of the mean as giving you a sense of the scale of the graph. In these real networks, there is no characteristic scale that you can describe the network with. And one kind of potential mechanism that they proposed would be one of preferential attachment. That is that you're not randomly placing connections between nodes, but nodes that have many connections are more likely to make new connections. So the analogy is with social networks. If you have a really kind of popular Johnny Hockey, whatever, you're more likely to become his friend than if there's some dweeb that nobody knows and that you don't want to be friends with. So this was their contribution at roughly the same time. Watson Strogatz published their classic network paper on small world networks. And they were playing around with this idea of what are the different extremes, what are the different kind of potential architectures that a network could take. And what they were doing was to start out with what are called ring lattices. So networks where the neighbors, spatially adjacent neighbors, are connected with each other, but there exists no long-distance connections, no shortcuts between different parts of the network. And what they started to do was say, okay, I'm going to take two connections at random, and I'm going to start randomly swapping them. And this starts to induce a lot of these long-range connections. And if you go all the way to the other extreme, you end up with a fully randomized network. And they were looking at two properties in this network. One was the so-called clustering coefficient. So the clustering coefficient is simply the number of completed triangles in a network. So here, for instance, you have two connections here. There could be a triangle here, but there isn't. Here, there's a complete triangle. And the intuition is very simple. So if I'm friends with Jack and I'm friends with Jill, chances are Jack and Jill are going to be friends with each other as well. At the same time, they were looking at things like path length. So the path length in a network is simply the shortest number of contiguous edges that you would have to traverse in order to get from one node to another. And the kind of the characteristic path length in a network is the mean of this quantity for all possible pairs of nodes. So if a network has a very short characteristic path length, it means it's very easy to get from one part of the network to another. So what they showed was that in a ring lattice, you have high clustering, but it's very difficult to get from one part of the network to another. You have to take a lot of hops. In the completely random case, you have these long-range connections, so the path length, the number of hops that you have to go through, is actually quite short, which is good. But this network is no longer clustered. And in between, there is this kind of broad regime of what they called small world networks that kind of had the best of both worlds. So they were highly clustered, as a social network would be, but at the same time, they had a relatively low path length. So just by inducing a few of these kind of long-range shortcuts, you could drastically reduce the number of hops that it would take to go from one part of the network to another. And this was sort of an analogy to Stanley Milgram's famous experiment about the 6 degrees of separation. So what these two papers showed was that there were, and actually this paper in particular showed that one nervous system, that of C. elegans, the nematode worm, actually displayed this kind of small world regime. So these papers collectively showed that a lot of real-world networks, both naturally occurring networks and man-made networks, all had very non-random properties, and it kind of spurred more interest in this field. So I'll just skip this for now. And so the reason why we care about network structure is because it can tell us a lot about how the elements work together and how the network is likely to function. So you can see this in many different applications. So this is, for instance, a network of transnational credit and investment. And the structure of this network can be used to predict kind of the onset and the character of bull markets and bear markets in financial networks. This is an example of a genetic interaction network, and it shows that genes that have genes with similar kind of interaction fingerprints tend to also be engaged in similar functions in the cell. And then, for instance, here, this is kind of a more recent development, this idea that people tend to follow on social media and promote work from other people who have similar political beliefs to their own. So more right-wing people tend to follow more right-wing literature, and more left-wing people tend to follow more left-wing literature, and there's kind of an increasing polarization of this network. So the reason why this is then useful to us is that the brain is a complex network that exists on multiple scales. So in terms of time, we know that brain structure can change over very short time scales. Certainly activity patterns can, for instance, due to learning, but then also on longer time scales over phylogeny, ontogeny, and so on. You can get changes in brain structure. In addition, as I kind of said, there's this kind of spatial scale where some people, as you heard this morning, study individual cells and how they're connected in specific circuits. Others, people who are more in human imaging, tend to study how entire regions are connected with each other and how how everything functions at the systems level. And finally, there is this other kind of topological scale where we are interested in how individual elements, so nodes and connections, participate in this network, how they can cluster together to form little functional motifs, little building blocks, how they make larger mesoscopic structures, and so on. So we'll talk about that as well. And the reason why this is pertinent is that in this day and age, we actually have lots and lots of ways to measure all of these things. So we can measure with very fine precision electrical activity, with slightly less fine precision hemodynamic activity. We're no longer studying just the brain, but really the entire central nervous system. Increasingly, there is literature on how brain rhythms are coupled, for instance, to gut rhythms. We certainly, and you heard from Adrian, for instance, this morning, how you can study the neural function and how it interacts with the environment of the organism. Some people study how brains interact with each other in a kind of a more social network kind of sense. So this field is really trying to build a mechanistic understanding of how neuron-level processes give rise to structure and function of sort of this large-scale system. So if you want to represent the brain as a graph, as a network of nodes and edges, the first two choices you have to make is how you're going to define the nodes and how you're going to define the edges. So these are questions that people spend entire careers on. So I'll just give a very simple overview. Essentially, the idea is when you're choosing nodes, you want to pick, you want to pool everything into one node so that there's relative internal homogeneity within that node. So you can choose nodes based on site architectonic properties. You can choose nodes based on various anatomical landmarks, so cell site gyri, things like this. Maybe you want to choose nodes such that everything in one node kind of has very similar functional properties, oscillates in a similar way, something like this. You could choose nodes based on receptor densities. Really, there are many different ways to do this. And similarly, there are also many different ways to do this with when you're actually placing edges. So I'll talk a little bit more about this distinction between structure and function, but basically, there are a number of invasive and non-invasive ways to reconstruct white matter connectivity or anatomical connectivity between areas. And there are also a number of ways to look at functional interactions between areas. So looking at either electromagnetic activity, hemodynamic activity, and so on. The basic idea is that we do something like this. If you have figured out how you want to divide the brain up into various nodes, you do so, and then using some histology or diffusion-weighted imaging, for instance, you can reconstruct the patterns of connectivity between all of those nodes. What you end up with is what's called an adjacency matrix. So it's another way to represent this graph. This is essentially a table where you have the nodes in the rows and, again, the nodes in the columns. And any one of these elements just tells you whether the two nodes are connected, and if so, how strong is that connection. So this is how you would derive a so-called structural network. On the other hand, if you're recording activity, you can record activity from the same areas. And here the idea is if I take two areas, and those areas tend to fluctuate coherently with one another, if there's some kind of statistical association between them, then we say, well, maybe they're engaged in some common function, maybe they're even exchanging information in some way. So we call that functional connectivity. And you can, again, do this for all possible pairs of areas, and you have a so-called functional network. So these are, not to belabor this point, but these are sort of two very different modalities. With structural connectivity, we're really talking about physical infrastructure, material connections, things you can kind of touch with your finger. With functional connectivity, we're talking about a statistical construct, patterns of interactions, and really, so this is, with functional connectivity, we're really talking about a system that can take multiple different configurations depending on what the organism is doing. Once you've gone through all that hard work, you now have a wide variety of tools that you can use to quantify the architecture of these networks. So this is what we'll talk about in the demo, but essentially there are a number of ways that you can quantify, for instance, the importance of a node. So the simplest of which is the degree, the number of connections that an area participates in. If an area has many connections, then it's well positioned to influence its neighbors and potentially the rest of the network. Some people look at things like motifs. So these little 3, 4, 5 node arrangements and how this potentiates different types of information processing in the system. So, for instance, if you have a very large number of these triangles versus these sort of open triangles here, this tells you about something about how the types of computation that network can do. There are various methods to measure or to quantify the potential for the network to integrate information. So, like I said, one of the most fundamental is the path length. You can then look at a whole range of things like what is the betweenness centrality of a node? What is the proportion of all shortest paths in the network that go through a given node? If a node sits on many shortest paths in this network, then it's well positioned to integrate information from multiple sources and also to disseminate information to multiple sources. One thing that I kind of hope to cover in the tutorial is this idea that the brain actually has a very prominent community structure. There are clusters, areas that tend to be spatially adjacent, that tend to have similar functions, also tend to be anatomically connected. And this clustering allows then for specialized processing, either visual processing or auditory processing in different parts of the brain. At the same time, there do exist a number of well-positioned high-degree nodes that tend to have connections not just within their own module or community, but to other modules and communities. And one very interesting non-trivial property that the brain has is that the high-degree hubs of the network tend to be densely interconnected with each other, above and beyond what you would expect on the basis of their degrees alone. So they form what's called a core or a rich club. And the idea here is that you have this infrastructure where, on the one hand, you can have segregated, specialized processing in different clusters, but at the same time, you have this highway that allows you to sample information from multiple specialized modules and to integrate it for whatever it is that you're trying to do. The nice thing is that right now, without doing any work, you can actually easily get these wiring diagrams for a wide range of organisms. So we have not complete, but somewhat complete information on various invertebrates like the nematode worm and the fruit fly. We have information on various avian species and various rodents like the mouse and the rat, non-human primates, and in the human with diffusion-weighted imaging. This is just more of the same. So I'll just kind of, for the last 5 minutes here before we go into the demo, just kind of give you an idea of what are the consistent results that we see across all of these different imaging technologies and tracing technologies and across multiple species. So the only, so far, nervous system that we have completely mapped out is the C. elegans. And so it's about 300 neurons, and in C. elegans, really it's the link between the anatomical network and various functional domains is quite clear. You essentially have a set of sensory neurons, motor neurons, and then somewhere in between them, interneurons. The sensory neurons tend to be kind of connected with each other. The motor neurons tend to be connected with each other, and The interneurons tend to receive a lot of projections from the sensory neurons, and they tend to send a lot of projections to the motor neurons. So what you have here is classic small world organization. You tend to have a high clustering, short path length, and this kind of heavy tail degree distribution indicating the existence of hubs. The hubs tend to be densely interconnected with each other, and they tend to span, they tend to sit in each of these specialized modules. And you can see that as well in the Drosophila here and the fruit fly. So there are various specialized communities that are densely interconnected that have to do with specific functions. Some have to do with, yes? So this is like genetic labeling in the Drosophila, for instance. So you... This is projection. These are all just actual neurons being traced out with microscopy in C. elegans. And in Drosophila it's a little bit different, but the way that I think you have, I'm not a biomolecular person, but my understanding is that you have a sort of GFP labeled viral vector, and then you put that into this is not layered cortex. It's just neurons tend to just kind of cells tend to clump together. So you just put a viral vector, and you can kind of see the expression of that vector along the axons. So here again, you have specialized communities. You tend to have areas that sort of transduce visual information or vibrations in the air, but then you also have areas that have to do with flapping the wings and so on. At the same time, and these are the nodes that are kind of shown with larger circles, these tend to be the hubs of the network, and they tend to be densely interconnected with each other. So they allow information to travel from multiple specialized communities. Similar things have been shown in the mouse. So this is the very nice data set from the Allen Brain Institute. And we have evidence of this as well in humans with diffusion-weighted imaging. So again, communities and dense connectivity between these communities via a small subset of high-degree nodes. These high-degree nodes, so I should just mention, this is kind of the very first time this was shown. This was like in 5 participants. Since then, obviously, this has been replicated many, many times with much greater numbers. What is the purpose of having this kind of centralized rich club structure? What we know is that the connections between these high-degree nodes, they tend to be relatively few in number, but they, on average, span much longer distances, physical distances, which means that the wiring costs, so the material and metabolic cost associated with these connections is much greater than with other connections. So why have them at all? It turns out if you look at the distribution of all possible path motifs, so you look at the shortest paths between all possible nodes in the network and you kind of trace them out, the most commonly occurring path motif is one that goes from a peripheral node, a non-rich club node, into a rich club node, into another rich club node, and then to a peripheral node. So it goes local feeder, rich club feeder, local. So the idea is that this central structure, even though it's extremely costly to maintain, it actually serves as a highway. It allows you to easily get from one part of the network to another with just a small number of steps. So these long-distance connections actually provide topological shortcuts. And again, consistent with the idea that they allow you to absorb and disseminate information in the network. So then just to summarize for the structural stuff, what we know in most of these networks is that we have a characteristic kind of small world organization with high clustering, short path lengths. We have also spatially contiguous communities that seem to suggest propensity for specialized processing. They are interconnected by these high-degree hub regions that are densely interconnected themselves, so they form this topological rich globe or core. Then just in the next couple of slides, just to prime you also for, this is everything that I've shown is for actual structure and anatomy. What do we know about function and functional interactions? There's going to be a talk later on about fMRI, so I don't want to spend too much time on this, but the basic idea here is that if you can put a person in a scanner, and you can tell them, don't do anything, just sit there and think, or not think. So this is the so-called resting state or task-free paradigm. And many years ago, a guy named Bharat Biswal noticed that if you look, you can take the time series, say, for an area in the somatomotor system, and you can look at its time course. You can then correlate this time course with other areas in the brain. And what he noticed was that even though the person was not moving, was not engaged in any kind of goal-directed behavior, just sitting there, there were very prominent correlations, not just in neighboring parts of the somatomotor system, but also contralaterally as well. So the entire somatomotor network was fluctuating coherently when the person was just sitting there in the scanner. And since then, people have done this for all possible pairs of areas. And this is kind of a typical resting state functional connectivity matrix. So this is a matrix of correlations between different parts of the brain while the person is sitting there at rest. This functional connectivity matrix also has a prominent community structure. So there are certain areas that typically have common function. They tend to fluctuate coherently. And so this gives us the so-called resting state or intrinsic connectivity networks, areas that, for instance, we tend to associate more with top-down cognitive control. They tend to fluctuate together. Areas that tend to be engaged in orienting attention to behaviorally relevant stimuli, they tend to fluctuate together, so on and so on. So if we have time, hopefully I'll show you how to derive one of these using community detection as well. And I'm just going to skip that, and just as kind of a theoretical summary, hopefully I've convinced you that neural organization can be fruitfully represented as a graph. This gives us kind of a whole language with which to quantify, to articulate different types of properties in the network. And that what I didn't talk about too much, but this has lots of applications in psychology, engineering, various health sciences, and so on, because it gives you descriptive measures. It can be used to address many different types of questions. If anybody's interested, there is a journal associated with this field, and this is, if anybody wants to, some people like learning from books. If anybody wants one, this is, I think, the best one on the topic. So, any questions so far? Yes. Oh, they've been set to 0. Some people do that. Yeah, so that, because obviously, trivially, the correlation of an area with itself is going to be 1. So I think they just set it to 0. And anything else, like I guess maybe you're talking about the fact that you can see kind of lots of white here, whereas here it's not quite so clear. As you're saying, I think that's just a visual thing. Yeah. But good catch. Okay, anybody else? Okay. So, if you look then in the data that was sent to you, I'm just going to start this up. Everybody can see? Great. So, the folder looks something like this. There is a MATLAB script called scpt_class demo. That's what I'll be working off of. There is also data, which you can load into your workspace. And everything that that we'll talk about is all the functions are in this folder here, the 2017 _115 BCT. I'll go through that in a minute. So the BCT is the Brain Connectivity Toolbox. This is a tool that was put together some years ago by Mika Rubinov and Olive Sporns. So if you Google it, the toolbox is online here on this website. I also in the slides included a hyperlink. So and I'll put the slides up on the Dropbox as well. The nice thing about this is that they painstakingly went through nearly every single measure known to man in graph theory, and they wrote it in MATLAB. So you have, for instance, here, this is the actual connectivity toolbox. You have, tons and tons of functions, but they all have the same kind of header style, the same usage style, and so on. So it's once you get going with this, it's actually very easy to work with it. So that's what that is. So if you just add a path, hopefully you're in the directory. If you just add a path to the Brain Connectivity Toolbox and you load in the data, you'll see that I've included 4 different variables. One is called SC, that's structural connectivity. One is FC, that's functional connectivity. Let's now just go back so we're clear what we're talking about with structural. I'm assuming that not everyone here does MRI. So with structural connectivity, we can do this in vivo in humans using the diffusion of water. So the idea is that if you have water molecules that are completely unrestrained, they're going to diffuse kind of all over the place, but equally likely in all directions. In other words, that diffusion vector or tensor will be isotropic. It'll be like a sphere. However, if a water molecule is restrained by something, say an axon, it can't diffuse all over the place. It can diffuse up and down the axon. So you'll have this ellipsoid, and that diffusion tensor will be anisotropic. So we use this information to infer how axons travel through the brain. Basically, this is kind of a Wikipedia level explanation. You start out, you have the different voxels in the brain. You start out in a gray matter voxel, and you say, okay, find me the neighboring voxel that has the most elliptical looking diffusion tensor. Once you found it, move over into that voxel. When you're in that voxel, move on into the next one that has the most elliptical one, and so on and so forth. Do this, but never make a turn that's more than x degrees, because that's not physiologically plausible. If you get to don't go into CSF, and once you get back into gray matter, you're done. So you can do this basically for all possible pairs of voxels, and you have these tractograms. This is called streamline tractography. So that's how we infer white matter connectivity with diffusion MRI. If you see this SC matrix is 1,000 by 1,000. So I've gone ahead and taken a diffusion weighted data set, and I've parcellated it into 1,000 nodes. So you can do something like this, figure image SC. This is what the diffusion data looks like. You'll notice that this is completely unusable, unreadable. What you should do is use a function like SPI. SPI gives you, when you have a sparse data set like this, it'll actually put a white everywhere where there's no value, and it'll put these nice blue points where there is a value. So you can see here that we have a network that's 1,000 by 1,000. So the first 500 nodes are in the left hemisphere. The second 500 nodes are in the right hemisphere. So you see that you have more connectivity within hemispheres than you do between hemispheres. This is usually a good thing to check. Now I've provided also a functional connectivity network. This is, like I said, done by having a person. This was the same individual who went into the scanner. They did nothing. We recorded changes in hemodynamic activity, and what you have in your MATLAB path is this functional connectivity matrix, which is also 1,000 by 1,000, and that looks something like this. So you can see that areas that tend to be close together, their time courses tend to be correlated with each other. So you see this very prominent diagonal related to the question that we had before. You also see these prominent, I don't know what the word is, they're not off diagonals. Whatever it is, you see patterns of correlation between homotopic areas. So left auditory cortex is not just correlated with areas around it, but it's also highly correlated with right auditory cortex. And that's why you see these strips here. You should always see these strips if you're doing something like this. You also see in the code that I provided, I have this kind of pathological need to make these matrices square. You can do this by typing axis square. Makes it look nicer. If you submit a paper that's if you submit a paper where one of your matrices is rectangular, chances are we'll think that you don't know what you're doing. So, making the data square is good. So anyway, now we're at this point. We have the structural network, we have the functional network. And we can get to the good stuff. We can calculate some things about this network. So now I think I'll probably just switch over completely into MATLAB. So I've done a few things here, like, before I go on. So I've also provided the XYZ coordinates for each of the 1,000 nodes that we have, and I've provided their short form anatomical names. So these come from what's called the Desikan-Kilani atlas. So for instance, somewhat descriptive names are provided. Some things that I like to do are to, for instance, get rid of the diagonals in the functional connectivity matrix that's shown here. I also like, for the purposes of the present discussion, I'll just binarize the structural connectivity matrix. So I'll set everything that's not 0 to 1, and everything that's 0 will stay at 0. This just goes back to Adrian's earlier point also that MATLAB is really perfect for doing matrix operations. So if you can get away with doing it, you should. So some of the things that you might want to calculate about your network is its degree. So I'll show you what in the Brain Connectivity Toolbox. Oftentimes for every measure, there are a couple of different versions. There's a version that you would use if you had an undirected network versus a version that you would use if you had a directed network. This is diffusion-weighted imaging. We don't know who's projecting to whom. We just know that there's a connection, so it's an undirected network. So the typical syntax is, you put in your network and at most 1 or 2 more parameters, and you get the answer. So the degrees, you might want to calculate the degrees of your network. You do it this way. You have how many connections each node participates in. You can also calculate something like what is the density of your network. The density is the proportion of connections, of all possible connections that could exist, that actually exist. So if we go back actually to the SPI thing, the SPI function, when we showed the structural network, actually showed us how many non-zero elements there were. In this case, there were 24,302. So you can always check that the density that you're getting, for instance, or any answer that you're getting in your network, that it actually makes sense. So for instance, here, the density function is telling us that it has a density of about 2.4, so 2.4% of all possible connections exist. You can easily check that. You can say, well, I have 24,302 connections, and the total number of possible connections is the number of nodes, that's n squared. That's not correct. There we go. So the number of nodes squared minus the elements in the diagonal, of which there are 1,000. So you can check that that this is correct. This is not correct. you can calculate something like the path length, but you don't. The Bring Connectivity Toolbox does actually this for you much better. Instead of calculating the path length between a particular pair of nodes, you actually get the entire distance matrix. So the distance matrix tells you for every possible pair of nodes what is, and I'll show you here, what is the number of steps, what is the number of hops that you need to take in order to get from one part of the network to another. You can see again that generally the low values are for nodes that are in the same hemisphere, and when you're trying to go between hemispheres, that's when you start to incur some of this cost. So that's all well and good. I think I had very grand plans that we would find a rich club and all of this stuff. I've included that code anyway, but what I think I'll do until I get played off is to just show you how you find functional communities in this network. So when we do community detection, now we're going to work with the functional connectivity matrix. What we believe is that some nodes tend to be more densely interconnected with each other than with other groups of nodes. What this comes down to is that you have your functional connectivity network, and you want to rearrange it so that all nodes that belong to community one